Hello, thank you for joining us. We are the UCLA South Bay team, and this presentation will cover the biodiversity assessment that we have been conducting at the newly acquired campus over the past year. So in 2022, UCLA acquired the former Marymount College in Rancho Palos Verdes. This campus is set to focus on environmental justice, climate, and sustainability. So as the site transforms into a hub for environmental education, our work helps ensure that its ecological potential isn't overlooked. By documenting and restoring native biodiversity, we're not just protecting native species, we're rebuilding local ecosystems and our connection to nature. So our work takes place at two main sites. The first is the 25-acre main campus in Rancho Palos Verdes, and 6 miles away, it's the villas where the students would be living.
We used two complementary methods to inform our final biodiversity report. Field surveys were conducted to identify the current state of biodiversity, and species distribution models were created to identify and evaluate potential biodiversity.
And for our field surveys, we went out to each site and assessed vegetation, invertebrates, birds, and mammals. We used a variety of different methodologies tailored to each group, and this was all just to get a baseline biodiversity at each site. And for our vegetation, we collaborated closely with Dr. Anthony Benyaga, who is curator at the UCLA Herbarium. And together we came up with two distinct transect-based methods for each site. And at the main campus, we ran 4 transects, and at the villas, we ran 2 transects. All right, and our transects at the main campus basically revealed that non-native species absolutely dominate the landscape. We found a total of 21 non-native species and only 1 native species across all 4 transects, the arroyo lupin. And at the villas, as you can see, native species were a lot more common. Our transects revealed that deerweed and California buckwheat were the most abundant, followed by coyote brush, which is pretty rare. And we only found California brittle-bush just walking around, but we did not capture it in our formal transects.
As for the invertebrates, we surveyed 16 different sites following research from Dr. Longcourt. This helped us detect both ground-dwelling and flying insects. So the specimens were then surveyed by entomologist Rick Rogers, which gave us a better idea of what we actually caught. So why do we care about this? Well, invertebrates are actually a great kind of a monitoring way for us to figure out what biodiversity is actually existing on a campus. What role do they serve? They're pollinators, decomposers, and even food sources. So as you can see right here, there's the abundance curves on both of the sides. And they're mainly dominated by pillbugs and earwigs, which are both disturbance-tolerant species. So this pattern reflects a very simplified urban ecosystem with a lot of room to grow. However, we did detect native species such as the Serratina and Perdita bees, which are both pollinators.
To collect data on bird species composition and counts, we conducted expert-led walking surveys and used the Merlin Bird ID app to identify birds from acoustic recordings. Our survey results showed that the main campus had more birds both in composition and in count than the villas. At the main campus, there were 17 unique species of birds, 8 species at the villas, and the 2 sites had 18 species of birds in common. Although this may be a simple sampling artifact, the results, at least in part, reflect the differences in habitat at each site.
So another very fun part of our project was the wildlife cameras. We deployed 6 different wildlife cameras across both of the campus to detect mammal activity. They existed in different habitats, such as a manicured lawn in the villas or an overgrown gully area in the main campus. These SD cards were periodically captured, and the species were identified. As you can see on the right side, there's an example of what our camera looked like, and there's a lizard on top of it that we unfortunately didn't capture on video. And here's an example of what the footage from the camera looks like.
Yeah, so our camera footage showed that the mammal communities are dominated by native urban adapters. At the main campus, the coyote and the desert cottontail were the most common mammals, while at the villas, it was the coyote and the striped skunk we detected the most. And a 1-week acoustic bat survey found 2 species of bats at the main campus and 3 confirmed species at the villas, including the western red bat, a California species of special concern.
And for our species distribution modeling results, we ultimately modeled for 136 species. And this is an example of what a spatially excluded map or model predictions look like. This one is for deerweed. Score closer to 1 indicates a higher suitability at that point. And for every single species, we extracted suitability scores at both the campus and the villas. And scores were usually pretty similar, as you see here with deerweed, but there were some exceptions. And using our model predictions, we determined that species with above a 0.9 habitat suitability have a high chance of success at each site. And this is just an example of what the unobserved species above that threshold, that 0.9 threshold at the main campus are. As you can see, a lot of them are native, and at the villas, this graph looks very similar. Now, using this list, we can make suggestions as to what species to include potential habitat restoration efforts.
So moving on to our discussion, we're going to start off with our limitations. So like most of the practicum groups here, our first is the time scale. We're only able to survey during the winter and spring months, meaning we missed the seasonal variation from a year-long project. Our traps may have also been biased towards mobile or larger invertebrates, as smaller ones might have been easier for us to miss. And then as for the cameras, overgrown vegetation was triggering false positive readings on the cameras, meaning we may have missed wildlife detections.
Restoring native habitat at both sites is one of our primary recommendations to South Bay administration. Our results showed that the main campus currently hosts little to no native plant diversity, but that it is able to support a large suite of plants typically found in native coastal sage scrub ecosystems. What we are working with is a blank slate, restoration-ready campus.
And at the villas, habitat restoration should center around the 4 native species that we found there: California buckwheat, California brittle-bush, the deerweed, and the coyote bush. We've already seen these species thrive there, and that indicates their potential suitability there and ability to support a healthy, resilient native ecosystem there.
We have also pinpointed 3 different threatened species that we hope to host on the campus one day. The first is the Palos Verdes blue butterfly, which relies on the deerweed and the rattlepod, which we aim to restore at the campuses. We hope to continue working with Dr. Jenna Johnson from Moorpark College in order to do annual releases and do pupae support. Our second is the coastal California gnatcatcher, which relies on a coastal sage shrub environment. We hope to have this at the main campus, which could then host up to 2 mating pairs. And as for the third one is the monarch butterfly that we so love. And we have spoken to the US Fish and Wildlife Service, and they have recommended that we plant nectar-rich native plants instead of something like milkweed, as this promotes healthy migratory behavior.
It's important to ensure that biodiversity is accessible to the students, staff, and visitors at UCLA South Bay. We are recommending that walking trails be placed through restored habitat, including plant signage, so that people can engage with and learn about the native species around them. And finally, this project is a work in progress. Our baseline biodiversity assessment was just the first step. Whether it's a future practicum project or something done by students at UCLA South Bay itself, long-term biodiversity monitoring is necessary to evaluate successful restoration and management practices, and it will keep future generations of students engaged in hands-on conservation work. Thank you guys for listening.
Questions from the audience about the incredible possibility for this property? Yeah.
For the stated goals, what were the factors you guys think will count as having
They use a wide range of topographic factors, soil, climate variables. And we trained a machine learning algorithm to predict the suitability.
Questions? Yeah, Noreen.
Wonderful job. This is definitely going to be an impactful project for UCLA. I'm curious on the plants, did you end up, so seeing just the one native plant or lupin, was this just transects of that hillside area, or did you also document separately cultivated plants on campus? Because I know there are a few natives like some buckwheat and ceanothus that are part of the campus landscaping.
Yeah, so for our transects, we did them in 4 areas. One was manicured non-native lawn in the sort of entryway of the campus. 2 of them were on the slope and then 1 was on the flat top area, so that's like the more undeveloped area. So within our transects, we only captured the arroyo lupine, but after the transects, we walked the entire campus with Dr. Maniaga, and we noted down some plants such as, it was not California buckwheat, but a different buckwheat that's also native, and lemonade berry as well. So there are some native plants such as those that we didn't capture them in our transects, but we know they exist on the campus, and those are included in our report as well. But at least when we did our transects, the only thing that we actually captured within our sort of plots was the arroyo lupine.
Great, thank you. Yeah, looking forward to reviewing the full report, and hopefully we'll be able to put the other project to work as we explore that station. Out at the type, see what fits and stuff.